HIV-1, also known as the 40-year pandemic. According to 2023 UN AIDS report, over 2.5 million people are infected with HIV each year, and more than 2 million lives are lost to AIDS-related illnesses. With the significant increase in access to AIDS treatment, we have successfully prevented approximately 20.8 million deaths related to AIDS over the last 30 years. This remarkable achievement highlights the importance of continued advocacy and support for these critical health services. The increased accessibility of AIDS treatment has allowed patients to stick to their prescribed medications, greatly improving the lives of individuals living with HIV. Despite ongoing efforts, complete eradication has not yet been achieved. According to statistics released by UN AIDS, approximately 1.3 million new cases of HIV infections were reported in 2023, and around 630,000 people died from AIDS-related illnesses. According to the updated WHO guidelines for the treatment and monitoring of HIV-1 in 2021, individuals living with HIV should not only take their medications but also undergo HIV-1 viral load testing using qPCR. This testing measures the amount of virus present in the blood and ensures that antiretroviral therapy is functioning effectively. Combining AIDS treatment with HIV-1 viral load testing allows for accurate classification of a patient's viral load status, enabling tailored antiretroviral prescriptions. This method not only helps extend patients' lives but also ensures that more individuals can access effective treatment, making it a critical aspect of care. Many low- and middle-income countries face considerable challenges in their medical infrastructure due to insufficient medical equipment and personnel, outdated facilities, and difficulties in service delivery. HIV-1 viral load testing is essential for effective treatment, but its implementation faces several challenges. We have addressed these limitations and developed an affordable HIV-1 viral load test kit. Bioneer's AccuPower HIV-1 Quantitative Real-Time PCR Kit received pre-qualification from the World Health Organization in September 2024, making it the first kit in Asia to achieve WHOPQ in the HIV NAT category. Meeting the WHO's strict quality standards ensures global product reliability. It is anticipated to play a vital role in supplying essential products for public health programs, particularly in low- and middle-income countries. The AccuPower HIV-1 Quantitative RT-PCR Kit is a ready-to-use solution that contains all the essential components for reverse transcription quantitative PCR. Each tube is pre-dried with reverse transcriptase, DNA polymerase, primers, probes, and DNTPs. This design eliminates the need for additional pipetting beyond loading the sample, providing users with a simple and convenient workflow while ensuring accurate and consistent results. Bioneer's AccuPower PCR Kit incorporates patented dual hotstart technology, enabling precise detection of even the smallest quantities of the HIV-1 virus. Bioneer's dual hotstart technology features two hotstart polymerization processes, a hotstart reverse transcription and a hotstart PCR reaction, which minimizes nonspecific amplifications at each step. In a standard reverse transcription PCR, nonspecific cDNA is produced due to nonspecific primer binding to a mixture of non-target RNA at low temperatures. However, the first hotstart reaction in the dual hotstart method eliminates the synthesis of nonspecific cDNA and significantly enhances the efficiency of the subsequent PCR amplification. In the second hotstart reaction, DNA polymerase remains inactive at room temperature but activates at elevated temperatures. This leads to highly specific PCR products, enabling the amplification of a small number of RNA copies. Excitation systems are designed to work seamlessly with all of Bioneer's AccuPower PCR kits. They automate the entire process, from nucleic acid extraction to PCR setup, once the samples are manually loaded. However, manual sample loading can lead to misloading and cross-contamination risks. The AccuLoader minimizes these errors by covering all sample loading tubes except for the one that is assigned automatically through barcode scanning of the specimen tube. For high-sensitivity diagnostics like HIV-1 viral load testing, it is crucial to prevent false positive results caused by aerosol contamination. The ExaPrep 16DX and ExaPrep 48DX instruments are equipped with a 4-layer patented protection system, which includes a droplet contamination shield, an aerosol generation shield, an aerosol protection cover, and a UV lamp. This comprehensive protection system significantly minimizes cross-contamination, even when processing high-titer samples. Bioneer's molecular diagnostic systems offer a simple 4-step workflow: sample loading, nucleic acid extraction, PCR tube sealing and loading, and real-time PCR. The first step, sample loading, is made faster and more accurate with the AccuLoader. The hands-on time required for kit loading is under 5 minutes, and test results are provided within 2.5 to 3 hours. In a comparative evaluation against a competitor's product, the AccuPower HIV-1 Quantitative RT-PCR Kit by Bioneer demonstrated outstanding performance, with an R-square value of 0.9595, sensitivity of 96.99%, and specificity of 100%. HIV-1. With proper diagnosis and treatment, patients can enjoy longer, healthier lives. Convenient and reliable testing for accurate monitoring of HIV-1 viral load. Bioneer is committed to driving innovation that prioritizes and elevates patient value.